Hello, dear colleagues. This is my 8th lecture. My name is Professor Sergey Shityakov. I am a head of the Cheminformatics Scientific Group at the Infochemistry Scientific Center, the ITMO University. In this lecture, I would like to give you some introduction on modeling of protein dimerization and folding. And first of all, I would like to give you some overview on the dimerization, why it is important in terms of the rational drug design and discovery. And this concept we are widely using in our project, in particular in chemoinformatics. The dimerization of the protein is the property of some proteins, such as viral proteins. On this slide, you see that some viruses have some proteins which are called the proteases, HIV proteases, and they have two parts, two subunits, which makes them the dimers. And these dimers represent, once they are combined, they represent in the middle, they represent the binding site, which is very important for the function of this particular protein. Therefore, if we will inhibit this particular binding site, we might interfere with the HIV virus life cycle, and this will help us to treat this very severe disease. At this point, we are going to implement different bioinformatics, chemoinformatics strategies to analyze the dimerization of the proteins in terms of the binding sites, and how these binding sites probably affected by either by the mutations, or if we will try to separate different monomers from each other, and how we can actually impair the binding sites. This model is also very widely used in computational chemistry, in a rational drug design, because this is the symmetrical binding site, and one could analyze it at very high accuracy, trying to implement new algorithms, such as on this slide. You see that some molecules like indinavir, they would tend to go to this particular binding site and inhibit the function of this protein. In our projects, we are using different parameters like the dimerization strength, when we have the information on the dimerization and how the monomers are bound to each other. So if we will separate these dimers, these monomers in a dimer, then we could probably understand at some point how the binding site is formed, and what is the function of the different parts of the binding site. And most importantly, if we will have the molecule which will bind to this particular binding site, that how the affinity would change if we would use different dimerization binding concept. On the other hand, we could also analyze different proteins in terms of the molecular folding, and the molecular folding is the universal property of the protein. The proteins might be completely folded to ensure the full function of the protein, because if the protein is unfolded, then the function is also lost. And at this point, the proteins should be folded to build up the the functional sites of the protein, either the binding sites of the protein, or different domains or motifs where the interaction is taking place or enzymatic activity is taking place. Now on this slide, we have the protein which is called the trip cage protein. It's the small proteins particularly useful for the analysis of the folding, for the protein folding. And here we are devising some parameters. We are using topological analysis, topological bioscaling analysis as the universal measure of the protein folding. And for this particular protein, we implemented some novel algorithms where topological properties such as dimensionality correlates with the radius of gyration, and thus by the adjusting these parameters, the molecular interaction can be also controlled. We investigated these correlations using the example of the well-studied short trip cage protein model. Additionally, we could integrate all this information, either the merization concept or the molecular folding concept, to our bioinformatics cheminformatics protocol in rational drug design and discovery to improve the pharmacokinetics and pharmacodynamics of different molecules, to understand protein ligand interaction, to understand host-guest interaction between different chemical entities, such as some drug-like molecules and hydrophilic cyclodextrins. The other is we can also implement these concepts to predict different physicochemical properties of proteins, such as solubility of different proteins, because if the protein is folded, it would also change the solubility. Next, we could also use these concepts in a materials design if we want to integrate different materials such as carbon nanotubes, fullerenes, morphs, and etc., to come up to a better drug-like molecules. We could also implement this information for the synthetic purposes, to use different enzymes or maybe to design some proteins with the superior enzymatic activities. To sum up, we could use for some cases like virtual screening that can be created for the new drugs, and study some proteins for the analysis of protein mutation and how they mutate and how their function is affected, which could also lead to the fact that the immune system might be at risk. Next, we could use these paradigms for the dependence for the projects where we analyze the conformational shifts in different proteins in order to create some drugs which are active in different pH media. So we might come up to the better substances to treat some very robust diseases. This is all in terms of the molecular folding, and dimerization concepts, and I thank you for your attention.